Yet another microbiology lecture. Looking at bacterial DNA and how it works. Kind of talked about replication, gene expression. So now let's look at regulation of gene expression. Again, getting into some of the nuts and bolts of it is kind of interesting because we actually can and understand it. Again, I'm not going to hold you to writing a lecture on the lac operon, but I do want you to be exposed to it as a great example of control. First, we have to do some background. Let's get some terms down so we're all on the same page. A little bit more review in this lecture, and then we'll go through the lac and trip inducible repressible operons, and we'll call it a day. So controlling gene expression in prokaryotes, again, producing a functional product is going to be our definition of gene expression. We want to remember that can be a protein or an RNA. We're going to talk a little bit more about details of how genes are sometimes arranged in prokaryotes that we don't see in eukaryotes, and that is arrangement in operons, where I can cluster multiple genes that are always needed together under one control element. Genes on the chromosomes and genes on plasmids are no different with regard to control, right? So wherever we see a gene, that's where we see a gene. Where we have DNA, that's where we have a DNA. And that promoter sequence is going to have its own kind of independent identity. So we can actually decouple a promoter or a control region from a gene and use it to control a different gene. So these are the ideas that are going to be the groundwork for biotechnology. So again, to reiterate, there's our bacterial chromosome, the plasmid. So especially as we start to build plasmids, we can see how we can cut and paste different pieces of DNA into a plasmid and then put that plasmid into a bacteria and literally change its capabilities. And so that's the idea with, again, this plasticity at a genetic level. That plasticity, kind of in a natural environment, is ultimately the result of natural selection. How can an organism respond to its environment? It's based on the DNA that has been selected for, right, through the evolution of that species. It works for that organism in its environment. And one level that we tend not to see so much in eukaryotes is this idea of phase variation. The fact that we've selected for genes that are randomly turned on and off. And you might think, how can that possibly be helpful? But the Fimbriae example is a classic one, for example, with E. coli. If you have Fimbriae, which I remember are pili, are kind of standard generic pili for attachment, if you have some portion of the population that at some random time turns them off, those can potentially tumble away to a new environment. Now, whether or not that environment is hospitable doesn't really matter because it's only some percentage of the population, but it may be. And so again, this is the idea of kind of the stochastic nature of evolution, and that there's no direction, but building the chances into the system for things to go right or wrong has been selected for quite strongly across all living things. And I think that is epitomized with bacteria in the sense that it's very willing to mess with its own DNA because every individual is 1 in a billion, right? The fact that I don't need to find a partner to reproduce if I'm a bacterium, the fact that we're so powerful and so good at making copies of ourselves through binary fission, I can just mess with anyone and be like, well, gone. No, as long as somebody survives. So that's a theme that we're going to come back to as we think about kind of this goop of DNA and moving between all the different quote unquote species of bacteria. I love it. So again, bacteria have to be able to sense their environment and respond. If you're a bacterium, it's complex behavior is very limited, right? If you could say any behavior is complex for a prokaryote. So the only thing that you really have is the ability to change your gene expression. And that can be in response to a signaling molecule. We talked about quorum sensing. That's a fine example of this idea. Some of you have studied signal transduction in eukaryotes. The analogous system in prokaryotes we call the two-component system. And surprise, surprise, along with the idea that eukaryotes are complex, prokaryotes are simple. Eukaryotic genes are more complex, prokaryotic genes are more simple. The two-component system is more simple than your standard, maybe multi-step, second messenger included signal transduction system in a eukaryote. Generally, this consists of a sensor protein, integral membrane protein, exposed to the external environment. It has an environmental stimulus that binds to it, that activates a protein on the inside of the cell, which then will result in gene expression. So, for example, a two-component system triggered by a nitrogen source. Nitrogen sources can vary. Any given one would have a different receptor. That receptor then activates a different activator inside the cell, which will then interact with the DNA to turn on the appropriate genes to process that nitrogen source so it becomes accessible to the bacterium. When we think about gene expression, we're going to break it up into a couple different levels, a couple different terms that we want to be familiar with. So you can turn a gene on, you can turn a gene off. So we talk about some constitutive expression, meaning that a gene's always on. We talk about basal level expression. So you can have a constitutive gene that is on, but at a basal level, that gene is going to be almost like a leaky gene. Like you just don't have a whole lot of expression, but it's there at some basal level. You can have genes that are inducible. In this case, the default is off, and something has to happen for it to be turned on. And then the opposite of that is the default of that gene is on, and then something has to happen for it to be turned off. Collections of genes then are often organized into operons. So again, think about metabolic pathways. So we've talked about glycolysis, we talked about the Krebs cycle, we talked about electron transport, and there are proteins managing all the intermediates in all those pathways. We haven't talked about specific pathways, amino acid synthesis, nucleotide synthesis, secondary metabolite synthesis. All of those are going to be managed by a series of proteins that hand one product, which has then become the substrate for the next enzyme, right? So when I need a pathway to work, I need all of those things. Why not just put them under the same operator, the same control element? And so hence the operon is seen. So what we'll see here is the structural genes are all lined up in front of the promoter region, and then there'll be a special part of that promoter region, which is often called the operator. We don't want to worry too much about those details, but you can look up greater level of detail with that. Somewhere else in the genome are going to be proteins that are going to bind to the promoter and operator, and those are the regulatory genes. Those may or may not be in an operon structure. Oftentimes they're not, but they will then have their own separate promoter that is going to be responsive to the environment to some extent. So let's talk about some examples here, and then we'll call it. So inducible genes, again, not routinely expressed, but they can be turned on when they're needed. So if you think about catabolic pathways, things that are breaking stuff down, and more specific than our foundational pathways that we talked about, but what do we feed into those foundational pathways? Well, we talked about glucose as the classic example, starting with glycolysis, but we also talked about how any of those intermediates can be a starting point. Well, where do those come from? I can break down other food sources, other metabolites to get to those intermediates. Or, for example, an alternative carbon source. So instead of using glucose, I'm going to use lactose. Well, if I'm going to break down lactose to feed it into one of those intermediates, I need enzymes to do that. But lactose isn't always around. So it doesn't make a whole lot of sense to maintain proteins which are expensive that I don't need. So these kinds of these specific catabolic pathways, and again, the example we'll use is the lactose operon, I'm not going to turn it on until I have the substrate that they're going to address, in this case, lactose. The opposite of that are repressible genes. So here they're routinely expressed, but then I'm going to have some mechanism to turn them off. And so here we think about some essential anabolic pathways, right? So the classic here is tryptophan. So I need tryptophan as an amino acid. So the enzymes I need to produce tryptophan, I want around, because I always want to be producing tryptophan because I'm always making proteins and I always need tryptophan in those proteins. One can, though, imagine, and we do see instances where that pathway gets ahead of production. And like, we have plenty of tryptophan around. Well, I don't want to overwhelm the cell with something I don't need right now. So I turn the system off when I have sufficient reservoirs of the target metabolite, in this case, tryptophan. So let's walk through this one more time. For example, in an induction situation, I have a repressor protein that by default binds to the DNA. So again, how do we say, oh, inducible genes are by default off, and then something happens to turn them on? It's oftentimes the case that a repressor protein is bound to the operator in front of the promoter, and RNA polymerase is physically blocked from transcribing those genes. What happens? An inducer, in this case, lactose, is introduced to the cell. The lactose has an affinity for a site on the repressor protein, and when it binds, it changes its shape. So think about allosteric binding, and then it falls off the DNA. When it's out of the way, transcription can now move forward. So again, the default is off. Why? Because there's a protein that is built to bind to that DNA, and I got to do something to that protein to change its shape so it gets out of the way. Right? Induction. Repression is going to be the opposite. I've got proteins floating around that their default is not to be able to bind to the DNA, but when a sufficient threshold concentration of, in this case, a co-repressor, in this case, tryptophan, for example, this amino acid that is the end product of the enzymes that this gene, this operon encodes for, reaches that concentration, it then binds to the repressor protein and activates it. And so now it's like, oh, we have plenty of tryptophan around. You just turned me on, so I'm able to bind to the DNA and stop transcription. This, again, is beautifully simple in the sense that as tryptophan concentrations fall in the cell, as it's being used up by protein production, that threshold you'll drop below the threshold. It will unbind from the repressor protein. The repressor protein then becomes inactive, and then RNA polymerase can once again transcribe the enzyme necessary to maintain a sufficient level of tryptophan. So this is a genetic control of a metabolite. Again, I can control that on an enzymatic level as well, but this just shows you a different level of control. And here's simply a more specific diagram of that repressible trip operon. So again, you can see at the bottom, the absence of tryptophan, the repressor dissociates, and RNA synthesis can proceed. So again, if I don't have enough tryptophan, I need to be making the enzymes that are going to make tryptophan. But when I reach that sufficient level, it binds, repressor binds, everything shuts down. Now, we can take this a little bit further and think about the lac operon specifically. So again, the lac operon is going to be inducible. A repressor by default is bound to the promoter, to the operator, and it's blocking transcription of 3 genes, right? So LAC-Z, LAC-Y, and LAC-A. Those are all going to be necessary really to get lactose into the cell and then be introduced to our metabolism so we can break it down and use it for energy. Right? When allolactose, which is just a slight version of lactose that the cell will modify to make, when it becomes present in the cell's like, ooh, there's lactose around. This binds to the repressor, it falls off, and now we're on our way to make all the machines we need to break down the lactose that we now know is present so that we can use it for energy. The fun thing about this is that we can control it on several levels. And so you can have the straight up, hey, is lactose here? I'm going to turn all these genes on and we're going to break down lactose and use it. Lactose is not present, so I'm going to block transcription of all these genes because I don't need them and I don't want to invest in machinery that I don't need. And that's great. And so you can work your way through these diagrams, and they're in every intro bio book as well. But what's really fun about the lac operon is that you can build it into a system that is part of preferential carbon usage. So what I can say is that, hey, this bacterial cell would prefer to use glucose. And if you give it both glucose and lactose, you'll see that it metabolizes the glucose first, and only when glucose is gone do you see the lactose machinery being produced, and you start to break down lactose, even though lactose is present the entire time. So if we delve into that, we can see a little bit more about how fun this can be, and it can be understandable. To do that, though, we have to introduce one more player, and that's an activator. So this goes back to this idea of kind of a basal level of transcription versus like an enhanced level of transcription. And so an activator is going to engage with the promoter and actually ramp up the ability of RNA polymerase to bind and transcribe an operon. And so in this case, what we're going to see is that only when glucose is not around is there an activator that engages with the lac operon to say, it's go time. Yeah, that lactose has been here the whole time. Let's really go after it. So thinking about how lactose then is available outside the cell, we have a couple different levels of control of it even getting into the cell. And so you can talk about how we control glucose coming in and lactose coming in, and how that can influence the state of the metabolism to ultimately lead to increase of cyclic AMP. And so basically what happens is when we have low glucose, we have pathways and mechanisms in the cell that will increase this molecule called cyclic AMP that we generate from ATP, and it's a second messenger, and it's going to bind to an activator protein. So this activator protein that we call CAP is going to then bind to the start of the lac operon and say, now we need to go. We're really going to make a lot of this. So if you look at this last slide, you can see that we've got our players. This is from a piece of primary literature, so the symbols look a little different, but you have your CAP protein bound with cyclic AMP. When glucose levels are low and lactose is available, there's no inhibition to the RNA polymerase, so it's going to be actively loaded, and you get a strong expression of the lac genes. If glucose is around and there's no lactose available, it doesn't really matter because the repressor protein is going to be bound because it hasn't been removed because there's no lactose around. If there's low glucose and low lactose, then our CAP protein is activated, but because there's physical blockage, like because the repressor protein is there, RNA polymerase is not going to get loaded. And then finally, if we have both around high glucose and high lactose, okay, so lactose is available, so the repressor protein is not there, but because we have high glucose, we don't see activation through cyclic AMP of the CAP protein, and so we don't ramp up the lactose genes. So we may see this, what I said, referred to earlier as a leaky expression. So you may see a little bit of it, but it's not going full speed. And we really want to be in a situation where it's like, ooh, lactose is the only thing we've got, so let's go for it, that we initiate that system. So don't worry about that too much, but having heard it is what's most important, because you may come across systems, biochemistry that will refer to this idea in the future. And so just so you may, oh, I can go look that up, okay? And then here's a chart that you can see that kind of summarizes that. It is a great kind of mental exercise to work through the lac operon. If you can do that, it literally will make your brain stronger. And so if you have the time, it's worth doing.